Thank you so much for the invitation to come and speak here today and to be the first speaker to really kick things off. I'm going to talk about somatic mutation profiling in cholangiocarcinoma. So since I'm the first speaker, I wanted to just give a little bit of background to make sure we're all kind of set in what we're talking about. So cholangiocarcinoma, cancer arising from the biliary epithelium. There are multiple possible sites. It can come from within the liver or intrahepatic, outside of the liver or extrahepatic, and in the gallbladder. These are aggressive malignancies with really dismal 5-year survival, and the majority lack identifiable risk factors. However, there is a significant minority of patients that are increased risk due to primary sclerosing cholangitis, liver fluke infestation, and biliary stones. And right now, there's really no effective screening and no really effective therapies other than surgery. So this just points out what a huge room there is for improvement in cholangiocarcinoma, both in diagnosis and therapy. So I'm a pathologist. I had to put in one picture, gross photo of a cholangiocarcinoma. So you can see we're in the liver, and then this kind of light sclerotic fibrotic mass is a hilar cholangiocarcinoma. So this is the enemy. This is what we're dealing with. So I wanted to give a historical perspective first. So what did we know about cholangiocarcinoma genetics about 2 years ago? Most studies focus primarily on identified cancer driver genes, particularly those found in pancreatic ductal adenocarcinoma, or PDAC. You know, this makes sense. Ductal adenocarcinoma is a ductal cancer. The thinking was that cholangiocarcinoma could be the same, so we'll see what frequency the drivers of PDAC have in cholangiocarcinoma. Unfortunately, many of the early studies didn't separate tumors by anatomic location, so the results are really hard to interpret. And then around 2 or 3 years ago, there was a study that found frequent hotspot mutations in IDH1 and IDH2 in intrahepatic cholangiocarcinoma. And to me, this was really the catalyst that said, these tumors are different than pancreas. We need to study them more extensively because they really don't just seem to be, you know, pancreatic cancer arising somewhere else. So what our group published a little over a year ago was the first whole exome sequencing study in sporadic cholangiocarcinoma, and we identified frequent inactivating mutations in 3 different chromatin remodeling genes. So what we did whole exome sequencing, so all the protein coding genes in the genome, in 32 intrahepatic cholangiocarcinomas. We did statistical analysis of these somatic mutations, and then we did a targeted resequencing of the potential driver genes in an additional 32 intrahepatic cholangios. In parallel, we did study gallbladder cancer. That was, most of you in the room probably know, these are really rare cancers in the US at least. So we started out with 9 gallbladder cancers, again did the stats, and then did targeted resequencing in another 8. And the biggest novel finding in this paper were really the inactivating mutations in the chromatin remodeling genes. So there were 3. Unfortunately, it's a little cut off maybe, but BAP1, ARID1A, and PBRM1. So I'll talk about the specific functions of these genes later. But each was mutated in somewhere between 15 and 25% of cholangiocarcinomas. But when you put them all together, almost half of the cholangios had a mutation in at least one of these genes. More impressive than the mutation frequency, though, were the types of mutations. So they were enriched for inactivating mutations, nonsense mutations that make stop codons, frame shift mutations, splice site mutations. So it really seems like these have a pattern of tumor suppressor genes, and these are function-altering mutations. So again, a schematic of the 3 genes, BAP1, ARID1A, and PBRM1. You can see the mutations, which are indicated below each gene. They're really spread throughout the sequence. You have missense, but you also have nonsense, splice site, and frame shift. So these have, you know, kind of the classic mutation pattern of being tumor suppressor genes. Each of these genes is actually known to be mutated in at least one other tumor type. So none of them are kind of new to the field of cancer genetics. BAP1 is in renal cell, uveal melanoma, and mesothelioma. ARID1A is in a ton of tumor types. It was first described in ovarian clear cell carcinoma, but it's in colon and stomach. And PBRM1 is also in renal cell. So what do these genes do? ARID1A and PBRM1 are both components of the SWI/SNF chromatin remodeling complex that, in a simple way that my brain understands, they kind of move the histones around so that the genes can be transcribed. BAP1 is a deubiquitinase that takes ubiquitins off of histones, but again is involved in regulating the epigenetic chromatin state and gene transcription. So this brings to mind, you have general mutations in very general chromatin remodelers. How do these then fuel tumorigenesis? And there are a few different mechanisms that have been proposed and studied in the literature. They might inhibit differentiation, they could promote cell cycle progression, or induce genetic instability, or maybe even other mutations. One interesting finding, though, that's come after our study came out is that BAP1 mutations actually seem to confer a significantly worse prognosis, so they may be useful as a prognostic marker in cholangio. One of the other findings that was not novel to our study but very striking were these mutations in IDH1 and IDH2. So in contrast to the chromatin remodelers that had frame shifts and nonsense mutations, all of the mutations in the IDHs were missense. And not only were they all missense, they were actually all at the exact same residue. It's kind of the classic pattern for activating mutations in an oncogene. Now, IDH mutations were originally found in glioblastomas. They've been described in other brain tumors and in some leukemias, but they had not been described in any other tumor in the GI tract other than intrahepatic cholangiocarcinoma. So cholangio is really different than pancreas. It's different than all the other GI cancers. So IDH stands for isocitrate dehydrogenase. It's a metabolic enzyme. Its normal function is to convert isocitrate to alpha-ketoglutarate. And the mutations that occur at the single hotspot actually make it convert to something else. It converts to this product called 2-hydroxyglutarate. And there have been studies that show that this new product of the mutant enzyme actually inhibits DNA and histone demethylases. Again, perhaps the mechanism for IDH1 is really epigenetic regulation. The story for IDH1 mutations and survival is still pretty muddy. Our study actually showed worse survival in the patients with IDH mutations. There was a big study of over 300 patients that showed longer survival in IDH1 patients, and then a more recent study that showed no significant prognostic difference with IDH. So maybe it has to do with who our patient populations are. It's still kind of not known what IDH means for patients' prognosis. The FGFR2 story in cholangio, I think, is one of the more exciting. We found somatic FGFR2 mutations, so we were doing exome sequencing, so these are small point mutations, but we found them in 13%. Importantly, some of these occurred at residues reported to be mutated in other tumor types, so we do think that they're probably functional. But several other studies, I started to try to update this list to get them all, but I couldn't even fit them on the slide. There have been many studies now that show FGFR2 rearrangements in cholangiocarcinoma, and I think our next speaker who's presenting an abstract is going to talk more about these, so I won't spend a ton of time. But FGFR2 is known to be rearranged to multiple partners in cholangiocarcinoma. They do seem to be associated with a better prognosis, but the most exciting thing is these seem to be sensitive to FGFR-targeted therapy, and these are drugs already in clinical trials. I mean, this is a figure from one of these original studies describing the FGFR2 rearrangements, and they actually treated a patient with an FGFR2 rearrangement with an FGFR inhibitor and showed actually tumor regression. That's really exciting. It's still a minority of patients. The prevalence is somewhere between 7 and 45%, depending on which study, but having a targetable therapy that's already in clinical trials is huge. So since our exome sequencing paper, there have been a few other papers that exome sequenced as well, and they found some genes that we didn't really highlight. One is the study by Gao and colleagues in Gastro last year. They did 7 intrahepatic cholangiocarcinomas, and they focused on mutations in protein phosphatases. So they found this gene called PTPN3 that's mutated in 41% of the cholangiocarcinomas in their series. They did some molecular biology and showed that the mutation seemed to be gain of function. I actually went back through our data. We had one mutation in this gene, but it was a nonsense mutation. So I think kind of the functional implications of these mutations aren't yet known. One thing to keep in mind, though, is this was a series from China, so it's possible that, you know, geographic differences, maybe the phosphatases aren't important in cholangiocarcinomas in the US, but in Chinese patients who might have different risk factors, they may be playing a bigger role. The ARAF protein kinase, which is in the RAF family, this again I think is going to be discussed later in the meeting, but it was in about 11% of patients. So there are other genes other than the chromatin remodeling genes that do seem to be important. So our study did look at a relatively small number of gallbladder cancers. We found that P53 was the most frequently altered gene in the discovery screen. It was about 60%, and it was the only gene with statistical significance. We did find infrequent mutations in the chromatin remodeling genes, a couple of mutations in BAP1 and ARID1A. PBRM1 was in about a quarter. But we didn't find any mutations in the IDHs in gallbladder cancer. Again, we used a small number of tumors. These are rare cancers in the US, so we can't make any definitive conclusions, but based on, you know, the preliminary data, it does seem like gallbladder is very different from intrahepatic cholangiocarcinoma, and this has been really confirmed in other studies that I'll talk about in a minute. So in the same issue of Nature Genetics as our study was this second exome sequencing study from a group in Singapore. And they did whole exome sequencing, but they compared liver fluke-related and non-infection-related bile duct cancers. So they did 8 liver fluke-related, 15 non-liver fluke-related cancers for whole exome, and then did targeting sequencing in a huge number of tumors, almost 200. So it was a huge effort. A couple of interesting things that they found in their comparison. So this is a comparison of all of the patients in their study. And they found that, they again found the chromatin remodelers. They found BAP1 and ARID1A. And they found that BAP1 was frequent in the non-liver fluke-related cancers, but pretty rare in the liver fluke-related cancers, whereas ARID1A happened in both. They found P53 and SMAD4, but these were actually more common in the ones related to liver fluke. IDH1 was much more in the non-infection related. And then just want to spend a second to look at GNAS and RNF43. Now, as was mentioned before, I do study pancreas as well. And these are actually 2 mutations that are in non-neoplastic cysts, or sorry, non-malignant cysts in the pancreas. So in pre-malignant precursor lesions in the pancreas. And they also showed up mostly in the liver fluke-related cholangios. And then finally KRAS, which was common in both types. But these data, this is just a summary of all of their data. They're not separating anything out by geography or by the site of the cancers. So if you look at just the Asian intrahepatic cholangiocarcinoma, so a much kind of pure comparison, you see BAP1 again is really just in the non-infectious cases. ARID1A is in both. P53 and SMAD4 are much more in the liver fluke related, whereas IDH1 is in the non-infection and KRAS is in both. So it really seems like liver fluke related and non-liver fluke related are genetically quite distinct. If you compare sporadic only but look at intrahepatic versus extrahepatic, again, BAP1 is really just intrahepatic, IDH1 is really just intrahepatic, whereas P53, SMAD4, and KRAS are much more in the extrahepatic. So extrahepatic cholangiocarcinoma genetically looks a lot more like pancreas cancer, whereas intrahepatic cholangiocarcinoma is really quite different. And Aldo Scarpa's group in Verona actually did a huge series of intrahepatic, extrahepatic, and gallbladder, and they found the same thing. They found, sorry, it's cut off, but this is BAP1 and IDH1 are really just in the intrahepatic, whereas KRAS and P53 are much more in the extrahepatic. So extrahepatic cholangio genetically seems like pancreatic cancer, intrahepatic seems very different. And there have been studies that actually show the more distal you get in the extrahepatic cholangios, the more they look like pancreas cancer. So kind of a summary of all the somatic mutation profiling on the individual gene level, about 50% of intrahepatic cholangiocarcinomas have mutations in chromatin remodeling genes, BAP1, ARID1A, or PBRM1. As far as the clinical implications, this is still kind of new and being worked on, but it does seem like BAP1 mutations confer a worse prognosis. 20% have hotspot mutations in IDH1 or IDH2. Because these are oncogenic hotspots, you could think of them as being a target for therapeutics, and I think those drugs are still in development because mutations in IDH1 haven't been known for that long. It's possible you could think of using them for diagnostics as well, but unfortunately these weren't in the liver fluke-related cancers, who would be the patients at highest risk. The data on prognostic implications for IDH1 is really conflicting. We showed it was worse, other people have shown it's better, so I think that the waters are still pretty muddy on that. FGFR 2-point mutations and rearrangements, though they do only occur at a minority of intrahepatic cholangiocarcinomas, they're perhaps the most exciting. They confer an improved prognosis, and most importantly, they seem to confer sensitivity to FGFR-targeted therapies, which are already in clinical trials. As far as the overall genetic landscape of cholangiocarcinoma, we can make some general conclusions. The altered genes do seem to vary by anatomic site. Intrahepatic cholangiocarcinomas have IDH1 and BAP1. Extrahepatic cholangiocarcinomas have P53, KRAS, and SMAD4. And gallbladder cancer, all that we found in our study was P53. They didn't have the IDH1 or the frequent chromatin remodeling mutations. The altered genes also do seem to vary by underlying etiology. The liver fluke versus the non-infectious do seem to be quite genetically different. Unfortunately, there's not much work yet on the other high-risk populations like PSC, stones, and choledochal cysts. And if you think about it, these would be the best patients to screen because cholangiocarcinoma is uncommon enough that screening the general population might not make sense. But patients with PSC or other high risks, if you had a genetic marker of people at high risk, it would be really useful. So I think that's a great task for the future, is getting these high risk tumors that arise in high risk patients and seeing what their genetics are. So with that, I'll just acknowledge the folks that helped out on our exome sequencing and follow-up studies. So Yu Chen, Tim Pollack, and Bob Anders were really leaders of different aspects of the project. Bert, Ken, and Nick are kind of the genome kings at Hopkins, and they obviously a huge help to me and everybody doing genome sequencing. We had a huge collaboration to get samples, both from Hopkins, Memorial, Mayo, Romania, even Italy. So we kind of huge multi-institution effort to collect the samples, and then Rachel Karchin and her group who did all our statistics. And with that, I am happy to take questions. Thank you very much. Sure. Of course. So it's hard to say, actually, because when you sequence cholangiocarcinomas, they're not very, oh, sorry, I should repeat the question for everybody who couldn't hear. The question was how often the inactivation is biallelic versus monoallelic in the chromatin remodeling genes. And it's actually hard to say because we're sequencing primary tumors and there's a fair bit of normal contamination. It does seem like a fair number of them are biallelic, but it's hard to say for sure just based on the amount of normal contamination in the samples. I saw somebody back behind. Oh, that was your question too? Okay, sure.
You suggested that there are ongoing studies looking at the implications of these. What specifically is being done to look at the therapeutic implications of these mutations outside of prognosis, from sensitivity standpoint?
Yeah, so I mean, we actually aren't doing anything. I think that one of the interesting points would be to look at, the therapeutic implications of epigenetic inhibitors like HDAC inhibitors, but I don't know if anybody's actually working on that. Yeah.
So based on your studies and also studies from other publications, seems that there's lots of diversity from the results, right? So what does it mean? Does it mean that it's more individuals or just different people are just simply different?
I think it kind of depends on if you're a lumper or a splitter. We talk about pathologists. You either try to lump everybody together and say they're the same, or split everyone apart. And kind of the lumper solution to that would be just to sequence more people. The splitter solution would be the reason that the data is muddy is we haven't figured out the right subsets to look in. So I think there's kind of room for both of those approaches. And I think the early studies in cholangiocarcinoma suffered because they just took all of them together in gallbladder and extrahepatic and intrahepatic. So I think trying to separate out the clinical groups and analyze the data could be helpful. But I do think there's room also for just sequencing more people, and also more people from more diverse geographies and things like that to try to find differences. Yeah.
Laura, very nice presentation. I think in addition to the controversy regarding the prognostic significance of the IDH mutation signature, as you know, there's also controversy regarding the pathological features of this particular signature. Being a pathologist, where do you stand? Do you see the IDH signature actually has unique pathological features, or you don't think so based on what you find and also the literature?
I have to admit, I haven't been super scholarly about reading every publication super carefully. In the tumors that we saw, there was not really a pathologic distinction between the mutant and the wild type IDH cases in our set, but I haven't read the literature very carefully to see how much I buy into what other people have done.
Andrew, could you educate us about what you know about pathological features?
Yeah, absolutely.
Yeah, so the only study from the Mayo group seems to suggest that with the IDH mutation, you seem to have the poorly differentiated histology, clear cell phenotype, but ours results, doesn't seem to suggest that, and also, a few other groups, also, don't seem to actually have this similar finding. So that's why I'm curious, for you being a pathologist, whether you have any insight here.
Yeah, I don't think we saw that in our cases. And again, the largest study that's looked at prognosis showed that they were better, that IDH mutations did better, and those being poorly differentiated to me, disagrees with that a bit.
I just wanted to make one comment that some work that we did actually with Daniela Sia, which looked at the gene expression correlates for the IDH mutant tumors in actually a couple of data sets, including some that we reported in our paper, suggested that they were distinct in terms of having a stem cell-like genetic signature or gene expression signature. My corollary actually to that is, are you trying to integrate overall genetic signatures and gene expression?
We haven't done that. We focus on DNA, but I certainly think there's room to do that, and multimodality is the key, I think. Yeah.
So,
oh,
sorry.
You listed a number of predisposing conditions for cholangiocarcinoma, and you didn't include hepatitis B and hepatitis C, which certainly they're the mixed tumors, but there are many patients with pure cholangiocarcinoma with chronic hepatitis. If you have a Chinese series, I bet you'll find hepatitis B in there. And
yeah, no, that's a great
point.
And
I think you actually said they arise from biliary epithelium. I'm not even sure that those tumors exactly do arise from biliary epithelium per se. I think that's probably going to have major genomic distinction that needs to be
yeah, I think the more correct statement would be to say that they have the phenotype of biliary epithelium. And that's something that one of my mentors, Ralph Ruban, would get on my case for having that come out of my mouth, because he always says you can't tell where anything arises, you can just tell what the phenotype it has currently. So I totally agree, and Hep C and Hep B are great points, and that certainly could be a reason for the difference in the Chinese study. Absolutely. Yeah.
Regarding the IDH1 mutations in gliomas, those appear to be the most clonal and deepest mutation in those genomes, and I'm wondering if the same is true when you look at the allelic index.
That's a great question. I don't think we've actually looked at our data with that question specifically in mind. One interesting point, though, that I don't think anybody's figured out the reason why is the most common mutation in gliomas actually doesn't occur in cholangios. And I always forget the specific amino acid changes, but it's at the right codon, but there's one mutation that occurs in gliomas, and it's like 80 or 90% of gliomas, and that one never occurs in cholangios. And I don't think anybody knows why that is. And mostly we noticed that because there's an antibody specific for that really common mutation in gliomas, and we tried to stain our cholangios with it and it didn't work. And it took us a while to figure out why that is, but it's that the amino acid change is actually different in cholangio.
Actually, I think probably Kate Yen from Agios will comment on that exact point.
Yeah.
But there is a clear different biochemical activity of the frequent mutation that's in cholangiocarcinoma, which is the R132C.
Yeah.
And it has a much higher catalytic activity than the glioblastoma prevalent mutation, which is R132H. So that's conspicuous that they have a different biochemical activity, and perhaps you need this higher production of the oncometabolite.
To work in cholangio,
yes, but I'm sure Kate will talk about that tomorrow.
Yeah. Yeah. Sure.
I was wondering if you have done any characterization of the normal surrounding liver for these patients compared to, let's say, PBMCs, DNA from PBMCs, to see if there's any sort of field defect.
You know, we actually haven't. It's a really interesting question that I think most people haven't actually worked on, is to know how diffuse these mutations are. Are they only in the tumors? Is there, if you do normal looking bile duct next door, but we certainly haven't done it. I don't know if anyone else has.
So I have to mention here that Eric Collison has an interest in that, and tomorrow there'll be a discussion session, and he has some interesting data to present about circulating DNA.
Interesting.
So when you were talking about the chromatin remodeling mutations, you made the point that these tumors are very different from pancreatic tumors, but then later on we hear about KRAS and SMAD4, and with the gallbladder tumors P53. So I'm wondering if you can take that, the tumors that have those mutations, and is there something clinically about them that would, define a subset that is more pancreatic after all?
It's a great question too. Unfortunately for the intrahepatics, I think we only had one with a KRAS mutation and one with a P53. So I don't think there's enough, at least in our study, to make any meaningful conclusions, because how do you know it wasn't just that person was 70 versus 50 or something like that. Certainly the extrahepatic seem to me more like pancreatic cancer, but as far as the intrahepatics, I think the numbers are so low that it's hard to really do much.
So this one comment, this has been a fabulous interactive session.
Yeah,
That's what we
absolutely.
want.
Yeah.
So Laura, as after your paper and several others like that, a lot of us try to almost routinely incorporate molecular profiling in our clinic, because there's a trial for IDH1 mutation, there's another one for FGFR mutation, and so on. But along with that has come actually more questions than answers, perhaps.
Just how genomic studies always go.
Right. So for instance, we find now with these reports, you get allele frequencies, you get combination of mutations. You know, you wonder what is a driver, what is not. For instance, you get a BAP1 mutation and you get an FGFR2 fusion gene. Do you have a very broad question in terms of how do you see the future of this mutational profiling in the clinic, and what kind of path forward there is in this sort of complex milieu of multiple mutations?
I mean, I don't think there's enough data to know exactly how it'll go. I think the important thing right now is to gather the data and use it in trials so we know kind of how to break out molecular subgroups and see who responds to what just with mutations. I don't think there's really a one-size-fits-all answer at this point. I think we're more at the data gathering stage, but I think particularly with the FGFR story, I think there is going to be room for targeted therapy, and the ideal cancer genomic targeted therapy model would suggest that, you have an IDH1 therapy, an FGFR2 therapy, and a BAP1 downstream therapy, and you treat patients with all of them at once so you get away from the emergence of resistance. But I think that we just don't have those targeted therapies yet.
Sure. Can I pose the same question to you, Funda, because you routinely, Funda is a chair of investigational therapeutics and it's kind of pioneered molecularly driven therapy at our center. How do you inform your patients and about treatment choices?
I think that's definitely Laura's thoughtful. We're not ready yet to make that answer. Is it the appropriate one? Clearly, I think we're going to start now targeting the oncogenes probably and try to understand the nuances of the chromatin remodeling and ultimately go to combinatorial approaches that will be more effective.
Yeah, and I mean, the question too is, as you mentioned, some people have two different mutations. Like if you have an FGFR2 and a BAP1, which one do you target? What mediates your prognosis? I think that's all still to be worked out as well.
Okay, if there are no other questions, we can move on to the next session. Thank you very much, Laura.